So we're from MIPS out in Parkville. So this is quite cool because we had Kate come and speak out to us about MAXIMA, so we thought we'd take advantage of this. So I look after all the imaging equipment at MIPS. So we have all the microscopes and flow cytometry and data analysis we do there. And most of the time I can write up code and get analysis done, but we kind of, with Dan's question, we end up with something much more complex that we hope someone here can either even just tell us the words to search for would be a great start, because we don't even know what we're really looking for. But basically, I think it's network mapping and everything else. But I'll leave Dan to explain the problem, and I'll chip in maybe a bit later with some of the microscope-specific issues.
Yep. Sounds good. Okay, so this is a slide taken straight from a lecture I gave recently. So basically, our research looks at the nervous system of the intestine. So within the gut wall, there is a neural network. It's formed of different types of neurons. These being sensory neurons, neurons that they couple to, and then neurons that actually make the muscle of the intestine do things or promote secretion by the intestine. This is an example here, basically an image of what's known as the enteric nervous system lying within the gut wall. Each of these dark dots are the nerve cells. And essentially we're doing live imaging of these cells, of these neurons, and looking at how these cells respond to a particular drug, and also how the cells that they interact with respond as well. Okay, so in order to do this, we basically take a piece of intestine, pin it flat, and then load it with a calcium indicator dye. This allows us to measure the changes in intracellular calcium. Okay, so in this image here, we've got one of these collections of nerve cell bodies, arrow pointing to a neuron. These are support cells known as glial cells. And also associated with these cells in the immune system. You can see they also respond, hopefully you can see this, to the drugs that we're adding. Basically, this dye allows us to measure which cells respond, the rate at which they respond, and also changes, for example, in oscillatory activity.
And also from that we can pull out the temporal and spatial locations of each of these cells. So we can ping each cell, tell you its individual response over time and where it is related to every other cell in the image. But the issue is then hopefully being able to turn that into something more sensible.
Exactly. Okay, so this is the sort of thing that we've been doing at the moment. This is a related video. So in this video here we have cells that respond. You can see that these cells have these sort of waves of calcium. If we take individual cells and then plot them out, you can see that there are cells that respond rapidly, sort of within the first 40 seconds, and then there are cells that respond later in the red. Okay, and they also have these oscillations. So in some way that these cells are interconnected. We believe that some respond directly and some respond afterwards through coupling to each other. What we have trouble with is how to present these data, how to analyze if there's any pattern between the interaction between these cells, and also basically a better way to present these data. So at the moment, this is not a pretty way to present data. It's a bit hard to follow. Other groups have done sort of similar studies, and they've done basically sort of a heat map: which cells respond, how long they respond, and sort of this is a demonstration of responses by this particular region here. You can see the cells that respond early, cells that respond later on, and then with a heat map, color-coded map over here. We're sort of interested in doing, I guess, something similar to that. As I mentioned earlier, there are a number of different cell types that respond within the tissue. We're interested in how to sort of show the interaction between these cells. Hopefully this plays.
No, it says codec unavailable.
The codec's unavailable. Basically, each 1, 2, 3, and 4 are these glial cells. You can imagine they're lighting up, they're responding nicely. We also have a blood vessel that was supposed to light up here, and also M, 1, 2, and 3 are macrophages. As you can see, the temporal dynamics and also the of these responses are quite different. So you get rapid responses, slow responses, sustained responses.
I guess part of the other question is too, do you get a certain type of response if cell type A or B is within a certain area of it only, or does it happen no matter what? And we can mark all these cells out and get all the information out about each cell and everything else without a problem. But it's more that complex, not just single cell type, but multiple cell types and multiple locations and everything else.
This sort of highlights that there are cells that do interact with each other. So in the purple is a particular type of cell that controls the nervous input to the muscle, and in the green are these macrophages. If we do a 3D rendering, see the green cell and the purple cell are closely opposed. We don't know if they're actually touching each other, but there's evidence that they do interact with each other in terms of signaling. This is an example here. Sorry. So with M we have the macrophage, and then I are these other cell types. And if we trace out the responses, the macrophage responds first, and then the other two cells will respond after that, suggesting that there's some sort of delayed interaction, perhaps release of something that activates the other cell type. We'd like to know, perhaps in a less biased way, whether there's a probability or likelihood of these cells responses being related, whether it's between these two cells are closely together, and also a better way to graphically represent this temporal information. Do you have anything else?
So can I just clarify, so the reason you want to understand about the likelihood of relationships between cells is so that you can infer those relationships and then kind of build models of this complex, tangled thing? Is that currently unknown?
Biologically, it's not known whether those cell types interact. What we are looking at may simply be, yes, this cell responds and that cell responds. Right. Whether or not the probability of these two things responding is linked to an actual...
But then being able to model it then means we can use those tissues and stimulate with various drugs, and we know we'll block one response or another and see if it actually does.
The goal is to build a model, but you can't build a model yet until you understand
how it actually works in the first place.
And you're using this observed data to try to infer those, relationships.
Yep. I mean, we can get other readouts as well. It doesn't have to be calcium.
It doesn't have to be calcium.
There's other pathways we can map as well using the same technique, but the data would be similar. A cell.
Yeah.
Yeah.